Okay, we're continuing with diseases of the microbial diseases of the urogenital system. These are the in the previous video, which was this is lecture 23, by the way, part 2. In the previous video, we looked at the bacterial infections, and here we're looking at viral infections. The first one I'll talk about is herpes simplex 2, or HHV2, human herpes virus 2. It's a virus that can cause a recurrent infection from viruses that this virus can remain latent within the nerves of the body and then come out, and it can recur. It is suppressed. In other words, you don't get, in some people, they don't get rid of it ever, and they have a latent, long-term latent viral infection. The virus remains sequestered within nerves and comes out with stress, fatigue, suppression of the immune response. The active infection itself can be suppressed by treatment with antiviral agents. I've named 2 here, but these are probably no longer sold. At least acyclovir is no longer sold. There are more recent ones. It can cause a neonatal herpes infection transmitted to the fetus or in newborns as they go through as they exit by vaginal birth. It causes it causes sores when it's activated. Another important viral infection of urogenital system is caused by some of the strains of HPV, or human papillomaviruses. Human papillomaviruses. The 2 that are of most concern are HPV 16, type 16, and type 18, which can lead to the development of cervical cancer, carcinoma of the epithelial lining in the cervix, and can also cause, in males, cancer of the penis. The only way to be able to tell if a person has a strain of infection that's with the type that causes cancer, these subtypes, there are many subtypes, is through DNA testing. But a much simpler way in avoiding all that is to get to be vaccinated against human papillomavirus. And these vaccines are available. They're against these 2 and 2 others that are associated with genital warts, and quite effective. Okay, now we'll move on to protozoa that can cause a sexually transmitted infection. In fact, it's the only protozoa that can cause sexually transmitted infection. This is Trichomonas vaginalis. Please do not mix it up with Chlamydia trichomonas. This is Trichomonas vaginalis. It causes trichomoniasis. It can be found in the semen and urine of male carriers, asymptomatic carriers of the infection. The vaginal infection with this organism can cause irritation and quite a profuse, that means plentiful, discharge. And diagnosis is by looking at samples under the microscope and directly identifying the presence of the protozoum in the sample. Let's move on. This is a yeast infection that can be a sexually transmitted infection, or it can be a yeast infection of the genitourinary system. It is found throughout the body. It's part of the normal microbiota in the body. It's on the skin and the mouth and intestinal tract, the genital urinary tract in the female. It's not in the genital urinary tract in the female. The male, usually everything gets washed out because it's all through, it's the ureter and urethra all the way down. It can cause a non-gonococcal urethritis. Remember, there is gonococcal urethritis caused by Neisseria gonorrhea, and then non-gonococcal urethritis caused by chlamydia. This is another cause of non-gonococcal urethritis, sometimes called NGU in males. In females, you can get a vulvovaginal candidiasis, an overgrowth of the candida. Sometimes it's due to fatigue, immunosuppression, poor nutrition. It can be also caused by excessive antibody therapy, which kills off competing bacteria so that the yeast overgrows. Again, treatment by antifungal agents is quite effective. Okay, the last few slides, I believe all of these last few slides, if I remember correctly, are all about a viral sexually transmitted infection, AIDS, caused by the human immunodeficiency virus. This idea about the syndrome or acquired immunodeficiency syndrome, and remember, syndrome is a set or collection of a set of signs and symptoms characteristic of a particular disease process. So this is a STI of the immune system. First came to the attention of researchers, virologists, and clinicians when there was a cluster of pneumocystis jirovecii infections and also Kaposi's sarcoma. It's an unusual, rare type of tumor cases. A cluster was observed in a group of several homosexual males. That was in the United States. Now, pneumocystis jirovecii doesn't normally cause infections in humans. It's all around us, but people are typically resistant to it and don't come down with an active infection. But it is characteristic, in fact, of a final end stage severe case of AIDS. In these individuals who have AIDS, they're severely immunosuppressed. Kaposi's sarcoma also is very rare and only was observed in this group. So they carefully studied the group and realized that they had a what appeared to be an infectious disease that severely damaged their immune system. And that was in the early 80s. Individuals showed a loss of immune function, and by 83, 2 years later, the virus that caused it was discovered. It's named human immunodeficiency virus. The virus itself is a RNA virus and has the reverse transcriptase enzyme, which allows it to copy its RNA genome into DNA, which can then insert into an infected cell. The DNA, viral DNA, can then be inserted into the infected cell. The cells that the virus infects all express a molecule on their surface called CD4. Now, CD4, remember, viruses have a particular host target cell. It's not just any cell in the body, but it's it'll be particular cells. And in this case, the virus targets cells in the immune system that express the molecule CD4 on their surface. Those cells are T helper lymphocytes and macrophages. So because it's a RNA virus that has reverse transcriptase enzyme, it's called an In fact, all those viruses are called retroviruses. The virus, of course, has the nucleic acid in the form of RNA, as I said. It has a protein coat around that made up of capsomeres, like every virus has this capsomere coat, and it is a enveloped virus. As you can see, it takes some of the phospholipid membrane of the infected cell, the cell it's infecting. When it leaves that cell, it buds out and takes some of the phospholipid membrane and uses that to form its envelope. And of course, there are viral proteins that are expressed in that envelope, and they're shown here. These are viral proteins. So the origin of the disease, acquired immunodeficiency syndrome, is that it is caused by HIV. It is a syndrome, a set of signs and symptoms. Probably, it's not exactly certain when, but it certainly seems to have crossed over from one species to another. So it crossed over the species barrier from primates, monkeys, in part of Central Africa to humans. Not an unusual thing for viruses to cross over to whatever species is eating them. A virus infecting a certain animal can often cross over into a species that eats that animal or is living in reasonably close association with that animal. And certainly in parts of Africa, the primates are considered, a hunting species that can be hunted and eaten. So probably occurring in the late 1800s or early 1900s, it's believed, with some good reason, to have crossed over. By going back through serum samples that have been stored away in freezer banks, which is not an uncommon thing at all, they have found the oldest known sample that is positive for the HIV virus, that has a virus or has antibody to it, was found in the serum of a patient who died in 1959 in the Congo, now the Democratic Republic of the Congo in Central Africa. It could have been there for an extremely long time. It's a bit unclear how long, but it could have been in human populations for a while, but not effectively have spread at all, hardly at all, just slowly infecting a small number. Until there was quite a large wave of urbanization and increased highway transport network built through Central Africa in the 60s and 70s. And then rapid spread due to that modern transportation and a change in sexual activity in terms of more prostitutes and prostitution with truck drivers, it's believed, transporting across long distances barriers, spreading the infection. The first case known in outside of Africa was a sailor who died in 76. So they go back on the samples, even though it was only first realized that this existed in the early 80s, they could go back on samples and discover some historical background. There are two major groupings, or what are called clades of the virus. HIV-1, which is more common and more virulent, much more easily transmitted between individuals, and HIV-2, which is seen more on the western extreme side of Africa. HIV itself, the virus, is as I said, an enveloped RNA virus, occurring by attachment of the virus to CD4 molecules on the host cell and then penetration. And that CD4 molecule is normally present and found on T helper cells and macrophages. What happens is that the receptor for that CD4 molecule will attach and allow the virus to attach to its envelope and then penetrate into the cell. And what you get is a typical kind of, and we've covered this before, a lysogenic kind of insertion of the viral genome into the host cell. So you get the virus attaches and penetrates, and then into the host cell, either a T cell or macrophage, and then using its enzyme reverse transcriptase uses its viral RNA to form a DNA molecule, which can then insert as a provirus into the host cell DNA. And the cell can live for quite a while and even divide, and it'll copy that inserted proviral DNA or provirus, and but eventually the virus starts producing RNA and RNA, its own viral RNA and viral proteins, and you get assembly and budding out, and often it goes also lysogenic and kills these cells. So and this occurs over a very long period of time. It's a slow process normally. What you have here is an active example of an active infection where you're getting expression of production of viral RNA and the viral protein assembly and viral proteins being expressed on the surface and budding out. They don't show the proteins being produced, but they are. And infectious viral particles being produced. And here you have the latent period of the infection with the proviral DNA buried, hidden within the infected cell. In the case of macrophages, they can act more as a kind of a reservoir. They have the latent infection there. The virus is in vacuoles inside, but it can switch over to an active infection where you get them being produced and budding off and getting released. So in terms of transmission and infection, it occurs through sexual contact, exchange of body fluid, and there are three typical stages. The first is asymptomatic, no symptoms or very mild symptoms. There can be a persistent lymphadenopathy, swelling in lymph nodes. The second stage is characterized by persistent infections with normally non-disease-causing organisms like Candida. I mean, candidiasis does occur in individuals if they're fatigued or tired or stressed or immunosuppressed or malnourished, but it's a fairly low grade but persistent kind of infections with normal microbiota. And then moving on, and this takes quite a bit of time, can take quite a few years, to category C, where you have full-blown acquired immunodeficiency disease syndrome and infections with typically CMV or tuberculosis, formation of tumor, Kaposi sarcoma, pneumocystis, girovecchi infections occur in very late stages of clinical AIDS. This is a diagram that shows the count, and this is typically what is monitored, the CD4 positive T cells. T cells have CD4 on their surface, at least the helper T cells do, and so they examine the T cell population of the peripheral blood for CD4 positive cells. Notice the time scale along here, it starts at weeks, but then these double lines here typically mean a change. What they mean is a change in the scale. And now you're going into years. Okay, so this infection, if left untreated, can go on for a long time and cause some drop in the T cell numbers, CD4 positive T cells. CD4 positive T cells just means T cells that have CD4 on their surface, and if they do, that means they're a helper type of T cell as opposed to the cytotoxic T cell. You can get some drop, but still there's enough to not be a problem, and then it gradually, eventually it starts sinking and sinking and sinking until the person is immunosuppressed because they don't have enough T cells, helper T cells. Meanwhile, if you look at a number of copies of RNA, HIV RNA in the plasma, how common can it be? How many can be found in the plasma? You get a early after infection, in the first 3 to 9 weeks after infection, you do get a spike. You do have this what's called acute HIV syndrome. Lots of virus, it's seeding itself in the lymphoid organs and infecting CD4 positive cells, but then it drops off. That's latency. You get into this latent period, and so you don't see any symptoms, but the virus is there. It is in cells, and it is infecting more cells, and it is killing off, as you can see. You get a drop off in the number of CD4 positive T cells until it gets to a stage where the T cell numbers, CD4 positive T cells, are low enough that you start seeing symptoms. And the person starts getting opportunistic infections in this period, and the count and the amount of HIV RNA present is much higher, and eventually you get to death due to chronic infection and weight loss and immunosuppression, etc. Now, all kinds of organisms are associated with infections with all kinds of organisms, or protozoa, viruses, bacteria, and fungi, all kinds of things, many of which never cause a problem in normal individuals who aren't immunosuppressed. All of these types of infections are associated with this syndrome. Also several different types of neoplasia, cancer, precancerous conditions. What you have in terms of diagnosis is very early on there is the appearance of antibody in the serum soon after infection. These antibodies, unfortunately, are not protective, do not get rid of the virus at all. So they can be used as evidence that the person was infected. That kind of what's called seroconversion, or conversion to presence of antibody, can actually take up to 3 months for following infection with HIV. And typically, in terms of screening for diagnostic purposes, they do look for antibody like this against HIV, and then they if they get a positive, then they confirm by looking for the actual antigens associated with the virus. That's the definitive diagnosis, by looking for antigen. Transmission is, as I said, by transmission, exchange of infected individuals exchanging body fluid, including through sexual contact, breast milk. You can, although often it doesn't occur, you can get transplacental infection of a fetus. Contaminated needles, blood-contaminated needles, sharing of needles between drug addicts, which is not uncommon, will cause transmission. Transplants of organs, artificial insemination, blood transfusion, although these are very, 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 very carefully examined before being used. Organs or semen for artificial insemination, or blood, they're all tested. But all of these other things can cause transmission from an infected individual to of the virus to someone from an infected individual. The HIV virus is always thought not to survive at all outside, but in fact it does. It does survive for some time outside of a cell. It can, not long, but this should say, it's kind of approximately a day and a half. Oh, sorry, inside a cell, this is outside a cell, about 6 hours, but inside a cell for a very long time, much greater than a day and a half. I'm not sure why I put this. It survives, it can survive for the lifetime of that cell, in fact. It could be years. Okay, I do have statistics to show you. I think they're up to current up to 2018. Here you see the worldwide prevalence of adult HIV. This is 2016 and this is 2018. Still a major problem in Africa, and and other areas, still a problem. And there are very few areas that are really free, but when you have 10% of a country's population, as you can see here, this last, the darkest box here represents areas that have 10% of the population of the country are positive for HIV. It's a pretty serious situation. Something to consider, huh? 5 to 10% or 10%, it's a huge incidence. Very serious. Here are statistics. I wouldn't hold you responsible for these. I just want you to have a clear idea of what's going on. In terms of people now, and I didn't mention it yet, but I will, and I'll start now. There are some very effective treatments to reduce the severity of the symptoms associated with HIV infection, so that people can live quite a long time. It is not without some side effects, certainly, but they are effective. But there is no cure. Once infected, the person will remain infected for the rest of their life and has to remain being treated, or will eventually become immunosuppressed because of loss of T helper cells and die of opportunistic infections. The end result of that is that as more people get infected, more are actually surviving and living with HIV infection. They don't, if they're properly being treated, they won't become severely immunosuppressed. They won't develop acquired immunodeficiency syndrome, but they need to keep taking their medication, otherwise they could. We're talking about quite a few million people and growing, and quite a few children actually in the world. Newly infected individuals are running at around up to, not quite, near 2 million new cases known, recognized, diagnosed new cases per year. The number of deaths, however, is dropping, which is why the number of people living with the infection is rising. 2016, still significant. I mean, 1 million dead in 2016, 940,000 deaths, 770,000 deaths. This is a website that gives data on infections, infection rates. In Canada, there's, in 2014 at least, 2,570 new infections. The breakdown in the new infections is as follows, with the group having the highest number of infections are gay and bisexual males. However, it is also a heterosexual disease without doubt. 33% new cases were among heterosexuals, and then 13 among individuals who also use drugs, who are sharing needles, essentially. New HIV infections in the US are up to, it's only 2014 data, but it's still indicative, quite a significant number. The statistics are a little bit different in terms of distribution, but nevertheless, it's a disease that can affect anyone, and anyone out there is potentially infectious, which is why you have to use caution. 2016 estimates 65,000 people in Canada living with HIV. And for Canada, new infections are in these kind of numbers, and as you can see, they are being reduced. Identification of individuals who are infected, proper information on prevention of sexually transmitted diseases is extremely important. Education, access to treatment, it's all of these are very important. Here you see the statistics off the website that I mentioned to you. Individuals with newly reported cases of HIV and cases of acquired immunodeficiency syndrome. You have to realize that they're having HIV does not necessarily mean that a person has AIDS. It takes quite a few years and no treatment to develop AIDS. So the number of cases of AIDS, and first recognized in 81, 82, so the statistics rose, and then with the development of some effective antiretroviral medication, it's been dropping off. But as you can see, new infections are still occurring out there every year. And the number of total people living with this disease is therefore the total number, these are new cases per year, so the total number of people living with the disease is rising and accumulating. Prevention by condom use, distribution of sterile needles. If they're going to stick the needles into them, at least give them clean ones. There's nothing wrong with that. They're going to do it anyhow. If they don't have clean ones, they'll use dirty ones, so might as well give them clean ones. Healthcare workers must use universal precaution approach to interactions with people. With infected people, you need gloves, gowns, masks, goggles. We used to recap needles after they were used and throw them out. They're not recapped anymore because that's the time when as you're putting the cap on, you could miss and stick yourself. So it's just discarded right away. No recapping. Risk of infection from an infected needle stick, accidental needle stick, is 0.3%. You don't want to do that. As I said, there are treatments, and there are a variety of them. I'm not going to go through them, but they have had a dramatic effect in reduction in mortality and a dramatic effect in preventing the development of the syndrome, the immunosuppressed syndrome, because will prevent the severe immunosuppression. However, and there are all kinds of quite sophisticated treatments, none of which I'm going to particularly describe, take quite a bit. But typically there are multiple different drugs that are used at the same time to try and avoid development of resistance to any particular one. And the key message, however, is that there is no vaccine. There is no effective HIV vaccine. As much as the effort has been huge to try and develop one, there is nothing. Treatments do reduce the viral load and do reduce the severities of the signs and symptoms, and therefore reduce the incidence of AIDS in HIV-infected individuals, but no cure. No effective prevention or cure, none at all. So it's something to always keep in mind. And with that, this is the end of lecture 23, part 2, and the end of the last lecture. And that's it, folks.